Good morning! It's April 11th, and this is your daily brief in science. Here's everything you need to know. A new breakthrough in the treatment of essential tremor has emerged with Fasical's Felix NeuroAI Wristband, which uses artificial intelligence to provide personalized therapy for this condition. This innovative device is currently under review by the U.S. Food and Drug Administration and aims to complement existing tremor medications like propranolol and primidone. The Tranquil study, a landmark trial involving 125 adults from the U.S. and China, is notable for being the first multi-center, double-blind, sham-controlled, non-surgical randomized trial in essential tremor to successfully meet its primary endpoint in over 2 decades. Results from this trial showed that participants using the Felix Wristband experienced a significant mean reduction of 6.9 points in modified activities of daily living scores, compared to just 2.7 points in the sham group. Both clinicians and patients reported improvements in symptoms and quality of life for those using the Felix device, with no serious device-related adverse events recorded, further reinforcing its safety profile. While some participants did experience mild skin irritation, the study confirmed that no serious adverse events were associated with the wristband. Dr. Rajesh Pawar, the principal investigator of the study, highlighted the urgent need for new treatment options for essential tremor, noting that existing medications often fall short and surgical options may not be suitable for all patients. The potential impact of the Felix NeuroAI wristband could significantly enhance the daily lives of individuals with essential tremor, enabling them to engage in activities that were previously challenging. Experts believe that AI-driven neuromodulation represents a significant advancement in personalized treatment, as it allows for real-time adjustments tailored to individual patient needs. Fasical, based in Minneapolis, is dedicated to revolutionizing treatment for movement disorders through innovative neuro-AI technologies, with the Felix wristband and other devices currently under FDA review. The findings from the Tranquil study were presented at the 2025 American Academy of Neurology meeting in San Diego, underscoring the significance of this breakthrough in essential tremor treatment. Researchers at University College London have made a significant advancement in the field of quantum computing. They have developed a groundbreaking method for fabricating quantum computers that boasts an almost zero failure rate and demonstrates strong scalability potential. This study presents the first reliable technique for arranging individual atoms in a grid, a breakthrough that took 25 years to achieve. This method is critical for building practical quantum computers, which have been a long-standing goal in the tech community. Previously, methods that utilized phosphorus atoms for positioning had a success rate of only 70%. However, the UCL researchers hypothesized that using arsenic could yield higher reliability, with initial results showing an impressive 97% accuracy in atom placement. The new approach involves positioning impurity atoms, specifically arsenic, within silicon crystals. This technique allows for low error rates and aligns well with existing silicon microelectronics technologies, which are already well established in the semiconductor industry. The silicon semiconductor industry, valued at approximately $550 billion, is poised to significantly contribute to the advancement of quantum computing, as arsenic and silicon are integral to semiconductor manufacturing. Quantum computers have the potential to solve complex problems more rapidly than traditional computers by utilizing single atoms as quantum bits, or qubits. Professor Neil Curson highlighted the milestone of achieving near-perfect precision in atom placement. However, he noted the need to overcome engineering challenges to automate the process for creating larger quantum systems. Currently, researchers manually insert arsenic atoms into silicon crystals to form a 2x2 array, which is a slow process that requires automation for scalability. Overall, this study marks a significant step toward the feasibility of building a universal quantum computer, with future developments expected to integrate seamlessly with current semiconductor processes. A new nasal spray vaccine for H5N1 avian influenza has been developed, leveraging technology initially created during the COVID-19 pandemic. Traditional vaccines for H5N1 primarily prevent severe disease, but are less effective at reducing viral transmission due to their intramuscular administration. This method generates strong antibodies, but fails to provide adequate mucosal immunity, which is crucial for controlling outbreaks at the virus entry point. The newly developed nasal spray vaccine can be administered in a single dose, effectively generating mucosal immunity. Animal studies have confirmed its safety and ability to produce comprehensive immunity, including neutralizing antibodies and T-cell responses. This advancement comes at a critical time, as genetic surveillance in March 2024 revealed mutations in H5N1 that enhance its potential to infect humans, raising concerns over possible human transmission and pandemic scenarios. H5N1 was first detected in humans in Hong Kong in 1997, and its capacity to mutate and spread presents a significant pandemic risk, particularly following a rise in cases in U.S. dairy farms that began in early 2024. If clinical trials for the nasal spray vaccine are successful, it could serve as a vital reserve for future H5N1 outbreaks, potentially shortening the timeline for vaccine development. In preparation for a possible bird flu pandemic, the U.S. government has allocated over $500 million for the development of mRNA vaccines. The study detailing the development of this nasal spray vaccine has been published in Nature Communications. Scientists at Rutgers University, New Brunswick have made a breakthrough with a new biosensor that leverages RNA to detect trace environmental chemicals crucial for human health. This innovative study, published in Angewandte Chemie International Edition, details how RNA can be implemented in bacterial cells to identify short-lived inorganic chemicals that are often challenging to sense. Led by Assistant Professor Enver Kagri Izgu, the research team employed a chemical mechanism where the binding of inorganic chemicals to a receptor produces light. This method enables the detection of substances like hydrogen sulfide and hydrogen peroxide within living Escherichia coli cells. This research builds on earlier findings from the year 2011, which demonstrated RNA's capability to bind small molecules and emit light. The implications of this new work are substantial, with potential applications ranging from monitoring environmental toxins to diagnosing diseases such as cancer, neurological disorders, and cardiovascular conditions. One of the primary goals of this research is to adapt the technology to convert human cells into sensor cells that can monitor disease-related chemicals without being rejected by the body. Izgu envisions a future where patients could provide cell samples that would be transformed into these sensor cells, facilitating real-time detection of harmful chemicals. In pursuit of this innovation, Izgu and co-author Tushar Agarwal have filed a patent application, with contributions from other Rutgers scientists, including doctoral students and a former postdoctoral associate. A new study from the University of Bristol is set to transform our understanding of brain development in young children, focusing specifically on how they acquire critical cognitive skills. This research tracks the development of 300 children aged 6 months to 5 years, building on data from the long-running Children of the 90s project, which has been monitoring their mothers since childhood. The Children of the 90s initiative has provided valuable insights for 35 years, following 14,500 children and addressing various health issues including obesity and autism. Recent findings from this project have established links between children's dietary habits and their social behaviors, alongside a troubling increase in fatty liver disease among young individuals. The current study emphasizes the importance of early intervention, noting that children who struggle academically in their early years are likely to face ongoing challenges into adulthood. Lead researcher Dr. Carla Holmboe highlights the need to understand the timing of critical skill development to better support children facing learning difficulties. Participants engage in scientific games and undergo MRI scans at various ages, which provide a detailed view of their brain development. In addition to cognitive skills, the research also assesses language development and processing speed, both essential for acquiring new information. For example, 2-year-old Jackson participates in a game designed to test his working memory, which is crucial for tasks like math and reading. Another participant, 2-year-old Henry, plays a game that evaluates his inhibitory control, where he must tap a screen to transform smiley faces into dancing animals while wearing a sensor-laden cap to monitor his brain activity. Emily, a participant from the original study, now brings her son Henry to contribute to the ongoing research, sharing her enthusiasm for the project's potential impact on future generations. Henry's game underscores the necessity for children to develop focus and adaptability in learning environments, as he must modify his responses based on changing stimuli. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.